All cells go through a cell cycle, which is a series of events that takes place in a cell as it grows and divides. The cycle has four main stages, starting off with the G1 phase. Cells spend most of their time in this phase because it represents the first chance for new cells have to grow. They spend about 10 hours carrying out normal cell functions. Next is the S phase. This is where the cell synthesizes its DNA. The last part of interphase is called the G2 phase. The cell has grown, DNA has been replicated, and now the cell is almost ready to divide. The last stage is all about prepping the cell for mitosis. The final part of the cell cycle is mitosis. This is the process in which a eukaryotic cell nucleus splits in two, causing two identical daughter cells. Interphase is where the cell spends most of its time before dividing. This is represented by the yellow highlights on the G1, S, and G2 phases. In this video, we are going to be taking a closer look at mitosis. Mitosis is defined as the division of a cell's nucleus, which takes 5 steps, and cytokinesis, which is the division of the cell's cytoplasm. The first stage in mitosis is prophase. During prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, condenses. The chromatin coils and becomes increasingly compact, resulting in the formation of visible chromosomes. The spindle fibers appear, and the nucleolus disappears. Moving on to the second stage of mitosis, prometaphase. During prometaphase, the nuclear envelope breaks down, and the chromosomes attach to the mitotic spindle. Both chromatids of each chromosome attach to the spindle at a specialized chromosomal region called the kinetochore. The third stage of mitosis is metaphase. During metaphase, the chromosomes line up at the metaphase plate, under tension from the mitotic spindle. The two sister chromatids of each chromosome are captured by microtubules from opposite spindle poles. In metaphase, the spindle has captured all of the chromosomes and lined them up at the middle of the cell, ready to divide. The fourth stage of mitosis is anaphase. During anaphase, each pair of chromosomes is separated into two identical, independent chromosomes. The chromosomes are separated by a structure called the mitotic spindle. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. The final stage of mitosis is telophase. During telophase, the chromosomes begin to decondense, the spindle breaks down, and the nuclear membranes and nucleoli reform. That completes the 5 stages of mitosis, but one more step needs to occur to form two complete identical daughter cells. That step is called cytokinesis. During cytokinesis, the cell membrane pinches in at the cell equator, forming a cleft called the cleavage furrow. A plant cell cytokinesis is the same concept, it just forms a cell plate instead of a cleavage furrow.